Hello, thanks very much for KDIGO and the organizers of this important meeting for inviting me to talk. I was asked to talk about kidney disorders with complex determination using FSGS as an example. My disclosures. So I'm going to try to at least touch on some of these points. What is a complex kidney disease? FSGS is an example of a complex disease. How can genetics aid with the diagnosis and classification of complex genetic diseases, and touch on some open questions and various priorities. So what is a complex disease? So this is a quote from the director of the National Institutes of Health. And he says, it's sort of funny that any disease would be called not complex. So this is one of those terms that initially seems a little odd, but in our own parlance, and geneticists have their own way of thinking about things, complex disease really is supposed to conjure up in your mind something that's not a simple Mendelian's single gene disorder. It's messier than that. He goes on, but it's somewhat of a vague definition. And perhaps as a working definition for today, we'll use something more like this. A complex disease we think of as a disease phenotype or trait that does not follow Mendelian inheritance patterns, and that's likely derived from multiple genes and non-genetic factors, and exhibits a range of phenotypes. So let's now talk about FSGS. FSGS, I think, is both a complex disease and it's a complicated disease also for a variety of reasons. First of all is just language, the way the phrase focal segmental glomerulous sclerosis or FSGS is used. Does this phrase really define a single biologically valid category? Probably not. So what do we mean by it? Do we mean, is FSGS defined probably as it should be by what a pathologist sees when looking at histology? Should we use it in the way that adult nephrologists typically use it in everyday parlance, the way pediatric nephrologists use this phrase? Are we talking about primary or secondary FSGS? Are those meaningful distinctions? And perhaps I would argue it's much better to split FSGS and recategorize things into etiologies defined by pathway, rather than simply the presence or absence of FSGS lesions on a kidney biopsy. When we talk about complex diseases, we're talking about genetic architecture, and we ask, what is the genetic architecture of the hopefully well-defined traits or diseases that we care about? What is the contribution of rare, highly penetrant variants? What's the contribution of common but less penetrant variants? And there's often the issue of phenotype. What phenotype makes sense to really try to dissect genetically? FSGS is a complicated phenotype. I don't think we all use this phrase in the same way. Again, if we compare it to, say, something else, another complex trait, let's say height. Height is much simpler. It's one number. CLCN5 related kidney disease may present what looks like FSGS on a kidney biopsy, but height, a 6-foot person is not going to present as a 5-foot person. FSGS, I mean, we make the diagnosis as if it's a binary trait, but is that even right? Does someone who has say 5 grams of urine protein and low serum albumin and edema, does that person have more FSGS than someone with, say, 2.2 grams of urine protein and normal albumin level? To really think of this precisely, we need to really define what we're talking about, or perhaps, at least from the point of view of genetic studies and genetic dissection, forget about FSGS as a meaningful term. So, as our knowledge of FSGS and related diseases, I will call them, progresses, you know, uh, this happens via a sort of iterative process. You know, our knowledge of these diseases has increased enormously over the last 20 some years. And in general, you know, we start when we in the process of learning, we start with some imperfect disease classification. We use that to learn something, and by learning something that we can then redefine the way we classify disease. So, for example, you know, there's what we FSGS histology in an adult kidney biopsy. There's what we call steroid-resistant nephrotic syndrome in children, congenital nephrotic syndrome in neonates. We now know, thanks to the work of a lot of people, that all these phenotypes can be caused by mutations in the potosin gene NPHS2 under a recessive model of inheritance. So that suggests maybe we should think about reclassifying this set of diseases. Maybe rather than thinking of these as three separate disease entities, we should call this, they're all part of the disease we call NPHS2 associated kidney disease, or at least maybe a slit diaphragmopathy. You know, once we know about NPHS2 and its contribution, we're better able to classify disease. We can say people either do or do not have NPHS2 associated disease. That improves our ability to find other genetic factors and further dissect things. So just to stick to the NPHS2 theme, you know, and again, was originally identified by positional cloning in families with apparent recessive steroid-resistant nephrotic syndrome. Then expanded studies of phenotypes and populations and families taught us that NPHS2 associated disease can present as congenital nephrotic syndrome, steroid-resistant nephrotic syndrome, FSGS. The age of onset is widely variable. Again, can present in the neonatal period, can present in adulthood. Mutations in NPHS2 account for some, perhaps 20 to 30% of steroid-resistant nephrotic syndrome. There's, you know, boatloads of references with giving numbers in this range, and depends, of course, on part on study design. We also know that, you know, NPHS2 we think of as mostly caused by rare mutations under recessive inheritance, but there are rare recurrent mutations like this R138Q mutation, and then there's at least one common variant which contributes to the disease, this R229Q allele, which has a frequency of about 3% in most populations, but only seems to be disease causing in combination with a certain subset of pathogenic alleles. There's a particular variant that seems to be a common cause of, or at least contributor to, nephrotic syndrome in Black South African children. It appears that people who are homozygous for this variant, R229Q, you know, this is not a disease-causing phenotype, but it might modify the effect of other genes. For example, in the large database known as GNOMAD, there are 186 homozygotes reported without any, presumably no history of nephrotic syndrome. And then there's a few reports that there are perhaps rare alleles in NPHS2 that act in a dominant manner rather than a recessive loss of function manner. So really, although we typically think of NPHS2 as a monogenic or Mendelian disease, you know, if we get into the details, NPH-associated disease, I think, you know, could be argued is a complex disease, and I think one could argue also that NPHS2 associated disease should be its own disease classification. It's all these entities share a common genetic basis, a common etiology, and probably, even though the spectrum of disease can be variable, they may end up responding to similar therapeutic approaches. So, we talk about Mendelian diseases and complex diseases. At some level, nothing is Mendelian, and at another level, one could argue everything is Mendelian. Nothing is Mendelian in the sense that even within, say, a single family with one clear disease-causing mutation, there are differences in age of onset and severity, and the pace of progression and the penetrance. The NPHS2, which we just talked about, is one such example. Here are two pedigrees with autosomal dominant INF2 associated FSGS. And the age of onset is highly variable. Some people progress to end-stage kidney disease, some do not. Some patients with, some of these individuals with mutations have no proteinuria or only minimal proteinuria. So really, monogenic diseases are often not really fully monogenic or Mendelian when you get into the weeds. On the other hand, I think some of us like to believe that ultimately everything is Mendelian in some sense. This is perhaps a little bit too much of a stretch, but we'd like to believe that every disease influencing variant ultimately has some binary effect on some biochemical or biological function, even if this is very upstream of the trait that we're looking at. So, if you had a stethoscope and somehow this stethoscope could say, I can tell by listening to your chest that transcription factor X isn't binding well to the promoter of gene Y, that would be perhaps a binary phenotype. And we think complex diseases are the collection of things which ultimately can be dissected into real biological effects. With what we call FSGS, there's this issue of phenocopies. So for example, UMOD, PAX2, CLCN5, type 4 collagen mutations have all been reported in people who were given the initial diagnosis of FSGS. So I think we have to be really careful about language and make sure we're talking about questions of medicine and biology and not questions of language when we talk about these things. So for example, if some people ask, do type 4 collagen mutations really cause FSGS? And I would argue that's not really a good question, because maybe that's kind of like a question of how you define FSGS. I think we can ask a better defined question, such as, do patients initially diagnosed on the basis of FSGS by biopsy have a different spectrum of type 4 collagen variants than those diagnosed initially with Alport syndrome on biopsy? That's a better defined question. Or is type 4 collagen associated FSGS phenotypically different from type 4 collagen associated Alport syndrome? We should not let our historical usage of FSGS lead to muddy thinking about these things. So back to complex disease. In typical parlance, we're generally talking about the sort of information that we get from genome-wide association studies. Many SNPs or other sorts of variants at many different loci each contribute a small amount to the phenotype of interest. It turns out that most of the common variants identified in most genome-wide association studies are found in the non-coding regions of the genome, that is in contrast to most monogenic disease. Humans, of course, have a wide spectrum of genetic variants, and these range from very rare to very common, and from things that have a hugely deleterious effect on phenotype to variants that probably have no biological consequence at all. And in general, recent rare mutations are more likely to have a directly deleterious effect on an encoded protein and are probably more likely to cause disease than any individual common variant. But it turns out that mutations, again, typically coding mutations, and expression regulating variants that are typically non-coding in the same gene, we now know can contribute to the same or similar phenotypes. So one example would be NPHS1, the the gene encoding nephrin, the important structural and signaling molecule of the slit diaphragm. So mutations in the nephrin gene and PHS1 cause congenital nephrotic syndrome under a recessive model of inheritance. That's a pretty monogenic disease. Many of the mutations associated with congenital nephrotic syndrome are clear loss of function mutations. However, recent studies now suggest that non-coding variants near NPHS1 that seem to impact NPHS1 gene expression are involved in childhood nephrotic syndrome and risk of childhood nephrotic syndrome. And in this study that I'm quoting here, a childhood steroid sensitive nephrotic syndrome, which we have traditionally thought of as a pretty different phenotype from congenital nephrotic syndrome. So what we learn about genetics is leading us to think about shared etiologies in ways that we wouldn't have been able to otherwise consider. So on the left here is this picture is found in essentially every textbook of human genetics. We think of, again, the spectrum from complex to Mendelian. Mendelian diseases are highly penetrant mutations that are each quite rare, and complex diseases are the effect of many common variants, each with small effects. And from this, in this nice review paper by Emily Grootman and her colleagues, they overlay a bunch of inherited kidney disease genes onto this kind of diagram. And on the upper left, you see, for example, TRPC6, INF2, ACTN4 cause a highly penetrant form of FSGs or nephrotic syndrome. There's some phenotypic overlap with other genes as indicated by this Venn diagram. And then on the bottom right are genes in which variants have been found to contribute to common forms, phenotypic descriptions of kidney disease like chronic kidney disease. UMOD is perhaps one of the best examples. And then ApoL1, you see, is somewhat of an unusual example. It's got pretty large effect, and these variants are pretty common. And we'll talk more about that as we go on. Increasingly, people are talking about polygenic risk scores. So essentially, taking the results of genome-wide association studies and then assigning individual people a risk score depending on the sum of predisposing and protective alleles that this individual has. And I think the verdict is still out on how useful this is going to be, at least as a clinical tool. But for some phenotypes, the polygenic risk score does seem to have as much of an impact on risk of developing that phenotype as do rare monogenic mutations. So for example, here's an example from a recent paper looking at, among other things, penetrance of BRCA1 associated breast cancer. So as you know, BRCA1 causes this autosomal dominant super high risk of breast cancer under a dominant mode of inheritance. And in general, people with these mutations or carriers have a much higher risk of breast cancer than people without these mutations. But on the left of this graph, you see that carriers who have a BRCA mutation on the background of a very low polygenic risk score have a slightly less, smaller odds ratio for developing breast cancer than people on the right-hand part of this graph, the people without BRCA1 mutations, but with a very high polygenic risk score. So an example by which, complex things are Mendelian things are complex. There's a tremendous overlap, and we need to think of, I think, most phenotypes in this way. And of course, there's, at some level, maybe that huge numbers of genes and huge numbers of variants affect almost every trait. There's this so-called Omnigenic model that's been proposed by Jonathan Pritchard and colleagues, and just to greatly oversimplify it, there's this notion of the core genes with direct roles in disease, and then peripheral genes, and these are essentially all other genes expressed in the relevant cell type. And at some level, many of these are clearly well-defined regulators of core genes, but the networks, gene-gene interaction networks and protein-protein interaction networks are so complex that almost any gene expressed in a given cell type may have some ultimate effect on the expression of a core gene. In general, in this model, the largest effect on phenotype is seen in rare, highly penetrant coding variants, as we see with monogenic or Mendelian disease. But for most phenotypes, most phenotypic variance is due to regulatory variation in these peripheral genes. So back to APOL1 associated kidney disease. So I think as most people know, APOL1 confers a very increased risk of various forms of kidney disease, various forms defined in sort of by historical phenotype definitions. There are two variants in APOL1 that are typically called G1 and G2 that both change the coding sequence of APOL1. Again, these are relatively common variants in people of recent sub-Saharan African ancestry. This is, again, somewhat atypical, the fact that these are coding protein changing variants, and are very common. If we look at, for example, the African American population, about 12 to 13% of this group of individuals has a high risk APOL1 genotype. So someone with a G1 genotype, a G2 genotype, or a G1 G2 compound heterozygote genotype. Those are the people who are at high risk of kidney disease. And we see a very large effect of having a risk genotype on the odds of developing certain phenotypes, and smaller odds for other phenotypes. For example, people with HIV infection in Africa, the odds ratio is 89-fold for the development of HIV nephropathy in the setting of HIV than if you have a high risk genotype than if you don't. And the odds ratios, maybe even higher for COVID-19 associated collapsing glomerulopathy. The number of reported cases is probably still too low to come up with a meaningful odds ratio, but to my knowledge, this phenotype has only been reported in people with high risk APOL1 genotypes. These odds ratios are somewhat less for FSGs, about 10 to 17-fold, and various ways of defining other kinds of chronic kidney disease. The odds ratios are somewhat less, down to, say, IgA nephropathy, where APOL1 variants seem to have no risk on the development of IgA nephropathy. So APOL1 associated disease is a complex disease, certainly, but with many characteristics of a Mendelian disease. And we should think of it as both, depending on the purpose of our thought process. So, for example, only about 20% of people with a high-risk APOL1 genotype ever go on to develop overt kidney disease. We think probably 80% of people with one of these high-risk APOL1 genotypes will not develop any kidney disease. So it's by definition complex, right? You need other things together with a high-risk APOL1 genotype to develop kidney disease. So, what are these other things? This is something that's being studied pretty intensively by a variety of groups. Is it one set of other things that leads APOL1, a high-risk genotype, to individuals to develop hypertension attributed CKD? And is it another set of other things that cause people with a high-risk APOL1 genotype to develop FSGS? Certainly we know that this collapsing form of FSGS, if we want to even call it a form of FSGS, the risk of developing this phenotype is greatly increased with certain viral infections, including HIV and COVID-19. So, what are these other things? Are they genes? Are they viruses? Are the genes which interact with a high-risk APOL1 genotype to cause hypertension attributed CKD, are they different from the genes and viruses that may interact with a high-risk APOL1 genotype to cause FSGS? I don't think we know the answer to that. So what are the clinical implications of all this? Well, we should certainly not expect FSGS to behave like a single disease or respond uniformly to any given treatment. I would argue that clinical trials in FSGS and associated diseases are likely to be much more successful if classified on the basis of etiology. And I think all of us people in pharma, regulatory agencies, academia, and patients really need to question and challenge trial designs that treat etiologically distinct diseases as if they were the same thing. I think irrespective of whether we look at FSGS histology or related phenotypes like steroid-resistant nephrotic syndrome or podocytopathy, we should regard these as complex phenotypes. And FSGS, I really think, is not a good name by itself to describe disease entity. And perhaps we need 3 or maybe more than 3 axes to classify someone with one of these diseases. We need to classify someone by pathology or morphology. What do we see on histology? What does the imaging of the kidney look like? What is the clinical phenotype? Is it nephrotic syndrome, nephrotic range proteinuria without the nephrotic syndrome, subnephrotic proteinuria? What is the, we have lots of words to define the clinical phenotype. And then what is the genetics? And I think we're always going to need all 3 of these things, and maybe even a 4th for environmental factors and other sorts of factors. But just as BRCA1 associated cancer is not enough to describe the cancer associated with BRCA1, we still want to know if it's ovarian cancer, is it breast cancer. Similarly, I think we're going to want genetics, but not just genetics to define these sets of diseases. I would argue that we should routinely be analyzing patients who are part of this FSGS nephrotic syndrome spectrum of disease for variants known to present with nephrotic syndrome FSGS or that have been reported to phenocopy FSGS and nephrotic syndrome. I won't get into the details of how we want to do that, whether it's by a large panel of genes, exome sequencing, but I think we should be moving to this notion of the default instinct should be to do genotype, not looking for a reason to do genotyping. Genotyping may affect therapy, for example, the drugs we choose to use, transplant decisions, and just as importantly, I think people want to know why they are sick. Doctor, why did I get this kidney disease? People have a right to know, and they're eager to know why they're sick. And so even if it is not immediately clear why making a genetic diagnosis is important, I think people like to know this. And of course, as we do this more and more, we'll collect more information and it will become, we hope, clearer how different genotypes will progress, how different genotypes will respond to different treatments. In particular, I would argue that we should be genotyping APOL1 pretty routinely. Almost 75% of African-American patients presenting with FSGS have high-risk APOL1 genotypes. Probably just under 1% of people who don't describe themselves as African-American in the United States with FSGS have high-risk APOL1 genotypes. And I think this is going to become particularly important because there are promising APOL1 specific therapies now in clinical trials. I think we should now be testing and be incorporating what we know already into clinical practice. For example, the relationship of genotype to steroid responsiveness, informing how we think about transplant decisions, risk to the donor, risk to the recipient, risk of recurrence. And I will end there. Thanks very much.